Hello, everyone, and welcome back to another episode of our Jack Westin MCAT podcast with your hosts, Mike and Molly. Over the last couple episodes, we've been really on a sort of sensation and perception kick, right? We talked all about vision, right? We also talked about hearing. We even talked about the physics behind that with waves. We really got into the weeds there a little bit with sensation, how all this works. But we've ultimately kind of left out a really important aspect of how this all works, right? Which is like, okay, we understand light is striking the retina. How does that activate anything? Like, how does that actually, how is that converted into a signal? And this is where it's important to talk about signaling cascades.
Ooh, super important. I'm so excited.
Yeah, it's going to be a good one. So, in this episode, we're going to talk all about signaling cascades, and we're really going to focus on probably the most important receptor that carries out these intracellular signaling cascades, which is a G protein coupled receptor or a GPCR. Now, because, we're focusing on GPCR, I feel like it's important to mention that there are other types. Okay, you might see passages that ask about or receptor tyrosine kinases, for instance, or something similar, right? You could absolutely be given other types of cascades and receptors in a passage. In fact, it's probably likely, but that doesn't mean that that's content that you are necessarily required to know. What you understand here about signaling cascades in general and about GPCRs can be applied to new contexts. So just to preface, you can see a lot more than GPCRs on in your passages, but an understanding of what we'll talk about in these episodes should be enough to get you through those questions.
Absolutely. And that's always like I think something that's so important to remind, from our whenever we have our conversations, like a lot of these topics, there's so much depth. Like a lot of times we only cover like what the MCAT topics, but then we talk about like electrostatics, for example, when we had an episode about that, like there's so much more actual depth that you need to have to do like a PhD to understand. We don't need all that depth. We're just gonna go on what the MCAT focuses on. Same thing here. Right, we don't want to kind of convey that GPCRs are the only, GP or membrane receptor that can cause a signaling cascade, but it's the one that the MCAT will expect you to have information on, that they'll expect you to have content on. RTKs, a really common example, is where it's something that you will see you probably will likely see in a passage, but the MCAT won't expect you to know about it before. And there's always going to be enough in the passage to answer questions on it. So I think that's always kind of an important, really important. So I'm glad you brought it up, Molly. Kind of context to go into this conversation. But coming back to like this kind of connection, right? I think we put it wonderfully where a lot of times we kind of like I don't want to say brush over, but we're like, oh yeah, once they receive it, it just turns into an electrical signal. When we got
Yeah.
it, and we're like, wow, that's the major thing. But I'm so happy that we actually get to dive in and be like, okay, what's actually happening here? What's actually going on so that you can actually turn this small thing into a huge response. And that's an idea that's really going to come back with when it comes to signal cascades. It's where we're turning a small signal, could be a single photon, could be a single molecule, into a large response in the cell. This is what we call amplification, right? Where we're going to talk about why and how these small signals can have huge responses in the cells when it comes to signaling cascades. And I think a great place to start off with signal cascades is the big picture, right? What is the overall kind of template for a signal cascades? Where does it start? And a great place to always remember, it starts with a signal. You need some signal to start. Without a signal, you're not having a cascade. Let's start there. Oh,
That's why they call it a signaling cascade.
no. So it has to always start with a signal. That signal can look wildly different. It could be light. It could be sound. It could be a molecule, right? It could look wildly different, but there has to be some sort of initial signal to kick things off. And that signal needs to interact with a receptor. And again, because we have a bunch of different signals, we have a wide range of different types of receptors. Now we have a wide range of receptors, and that receptor needs to cause some sort of a transduction or a conveying of the signal to something else. So that's why a lot of these receptors will be coupled to something else. So there'll be something attached or associated with that receptor in order to do the function. Many times, when outside of signaling cascades, receptors themselves do the function. We talk about like ion channels and that sort of thing. But for the conversation of signaling cascades, a lot of the times there's this protein coupled with the receptor that does like a lot of the brunt of the work. That's what we call transduction, where the receptor kind of conveys the message to this other protein. And it's this protein that leads to the amplification, kind of a pathway where one signal leads to two, two leads to four, four leads to 8, 16, 32, and you get this wide amplification, which leads to a response. However, we can't forget, but once we have that response, you need a way to end that response. So that's where always a signaling cascade needs to always end up, where you have a response, but as in all things, you don't want things on all the time. You need a way to turn it off. So we'll talk about termination of the signal after you have the response. But yeah, that's kind of like the universal template, you could say, of a signaling cascade, no matter the cascade.
And like we've mentioned, there are so many different variants of this, but I think really the unifying theme is there's a signal outside of a cell. It needs to get inside of the cell to carry out whatever the effect should be, right? There needs to be something that can take the extracellular signal and convert it into an intracellular response, right? If that signaling molecule cannot enter the cell, especially, which we'll talk about, it has to have something that kind of translates or transduces that signal inside, right? So yeah, that workflow, that general template is perfect. And I think at this point we're ready to think about G protein coupled receptors specifically. Again, this is really the most tested receptor type. We want to know a fair amount of information about it. And how it actually works. A lot of our signaling cascades that we talk about are done through GPCRs, which we'll talk about. But let's start off with just like, what the heck is this? Okay, GPCR is a G protein coupled receptor. So, like you said, this is a receptor that is coupled with another protein, namely a G protein, which is actually going to have a couple different subunits. Okay, if we think of the receptor itself, okay, let's go through this stepwise, right? The receptor itself is a transmembrane receptor, so it's embedded in the plasma membrane. It's got 7 alpha helices. Do you need to know that? Maybe not, but it's cool. It's the actual physical structure of it. Its job as a receptor is to bind a ligand that does not cross into the cell and to activate a G protein. Now, the G protein is going to be a trimer, right? It's got 3 different subunits, alpha, beta, gamma. Nice and easy naming. And before anything happens, right, when this is still inactive, they're going to be all snuggled up next to each other and next to the receptor. Okay, so all 3 G protein subunits and the GPCR are all connected, right? They're all in a large, call it a complex, right? Now, of these 3, alpha, beta, gamma, alpha is really what we care about. Not to say beta and gamma do nothing, but for our purposes, really alpha does all the action, alpha action. And it does this by, well, we'll talk about this in a moment, but whether alpha is doing anything special or not depends on what it's bound to. Okay. And one thing that I want to clarify early on, before we even officially start the cascade, right? And we talk about what happens when the ligand binds, what I want to clarify is these G proteins are named because they are GTPases. Okay. Which means it's an enzyme that cuts GTP. Okay. If that is the case, do you think it's going to be more active when it has GTP or when it has GDP, Mike?
I'm thinking about GTP is probably like, I'm thinking about that molecule with the energy. So it probably wants to have the molecule with the energy in order to do its job. So I'm thinking when it's GDP, not a lot of energy. When it has GTP, that's when it's doing its work.
Yes, exactly. So when you are inactive, the alpha subunit of the G protein, which is really what's going to go off and carry that signal out, right, throughout the or the inside of the cell, when it's inactive, it's going to be bound to GDP. Again, it can't carry out its function because it's already bound to GDP. Okay, this is important to know because a lot of students are like, oh, G proteins, they're probably going to phosphorylate it, right? This is all phosphorylation, it's kinases. There may be kinases downstream of it in the pathway, but really, when we talk about activation, we're taking that GDP bound to the G alpha subunit and we are replacing it with a separate molecule of GTP. Okay, so in just describing the general structure, I feel like it's really important to clarify what this is and what it's not. Okay, a G protein is a GTPase, which is how it gets its name. It's inactive when it's bound to low energy GDP, right? Diphosphate. And it's going to become active when that ligand binds and it exchanges for GTP. So now that we've laid that groundwork, Really, I mean, I feel like this stepwise process is it's pretty short and sweet, right? It's not nothing too crazy now that we've got that. So walk me through what happens when the ligand binds? What are the downstream effects?
Great question. So ligand's going to bind to the receptor. I actually always love making this distinction that the receptor itself isn't the G protein. A lot of people like
Yes.
look at the receptor and they're like, oh yeah, that's the G protein. No, no, no. The receptor is coupled to a G protein.
Yes.
So the ligand's not binding to the alpha, beta, gamma. No, no, no. Ligand's nowhere near
Mhm.
there. Ligand's binding the receptor on the outside of the cell, which is causes a conformational change to the receptor, which conveys that change saying, hey, the receptor kind of goes to the G protein, which it's attached to, and say, hey, something bound to me, you've got to change. Let's actually get things going. So that alpha beta gamma subunit, once the receptor changes in conformation because it's bound to the ligand, now it has that GDP on it and says, okay, I need to actually do something. And so a common kind of misconception is that, oh, it phosphorylates the GDP, turns it from GDP to GTP. No, no, no. It kicks off the GDP entirely and replaces it with a whole new GTP. So it's not like this reverse kinase situation going on. No, no, no. It's kicking off the GDP and adding a new GTP. And once it does that, once it's being told by the receptor to do that, that's when it becomes active. But now we have to recognize, what does it mean to be active, to be a G protein? Well, we talked about before, those alpha, beta, gamma subunits were all together, right? That was its inactive form. Well, in its active form, that's when they're going to come apart. Specifically, the alpha subunit. I love that kind of connection. Alpha means action, like that's the one that's doing the action. Big important to note that, like, yes, beta and gamma do things that are important to the MCAT doesn't really focus too much on beta gamma, kind of gives all the love to the alpha subunit generally, where they're going to dissociate. Alpha is going to go on its own, and that's the one with the GTP on it now, and beta gamma is going to go on its own. Right? It's this alpha, so now they've dissociated from one another. That alpha subunit of the G protein now has a few different options depending on the pathway that we're talking about, right? The main pathway that the MCAT's going to focus on is the adenylyl cyclase pathway, where that alpha subunit of the G protein's going to go, and there's this membrane protein known as adenylyl cyclase. Again, that cyclic part is going to be important in just a moment, right? And it's going to target that. However, we have to recognize it's not the only target that it can have. Depending on the pathway, depending on what that signal and receptor were, it could also target another effector known as a phospholipase C. Again, we'll dive a little bit deeper in a moment on these two different pathways. But one thing I want you to take away is there's only one option. There's a major option the MCAT will want you to know, like they'll want you to know these two pathways, that these are two effectors. And it's not just one. Then, well, we'll go on and we'll dive in more on these. But once it's actually targeted these things, again, this activated alpha subunit has targeted these things, these other enzymes are going to have their own impact, their own creation of messengers. This is what we call the secondary messenger molecules, right? The alpha subunit of the G protein was kind of like was the G protein itself. It was kind of like I wouldn't even say the primary messenger, but it was kind of the transduction of the signal. But these targets create what we call secondary messengers, where one alpha subunit of the G protein can cause the creation of a number of secondary messengers from that adenylyl cyclase, or it could target multiple adenylyl cyclases, which all create their own secondary messengers. This is where we start thinking about this exponential growth of the response, where one molecule led to a bunch of secondary messengers produced. This is what we hear when we think about amplification, right? Where it's like one molecule leads to two things produced. Like one secondary messenger leads to two other effectors. Oh, that leads to four effectors, then eight effectors, then eight, and that exponential growth. This is where we're actually seeing that amplification in the secondary messengers produced.
I think, I always think about early cleavage stage, like right after you have fertilization, which by the way, we just covered in an episode maybe like a couple weeks ago. Like early embryogenesis, all these good things, right? You have fertilization occur, right? You have a little zygote, sperm and egg have already met, and then it just divides and divides and divides and divides, like so many times without growth, right? But you recognize that if you have one cell and you cut it in half, you've made two. Each of those cells cuts in half again and you make four. Each of those cells cuts in half again, and so on, right? That's what we mean by exponential growth. I think sometimes it's easier to visualize this in a cellular context because we can imagine what happens when you just keep cutting everything in half. You make so many more than you would have started with. Same thing with, PCR with amplification of in that case DNA sequences. But, this exponential growth of the response is really key in helping us understand how G protein coupled receptor pathways work, right? And I think it even calls back to what we had talked about in a different earlier episode, which is on endocrine, right? Because we talk a lot about hormones and hormone signaling cascades. And when we think about GPCRs, these really apply to one and a half hormone classes, right? These are hormones that do not pass into the cell, right? If they don't pass into the cell, it's typically because they cannot literally pass through the plasma membrane, right? Plasma membrane, we know the interior is mostly hydrophobic, right? Nonpolar with all of those fatty acid tails. So typically, this is going to be our peptide hormones, which are typically water soluble, and most of our amino acid derived hormones, really with the exception of T3 and T4. If you're hearing this and you're like, huh? What is any of this? Go check out our endocrine episode. But a lot of our hormones, for example, that do not cross into the cell, as compared to like a steroid hormone, for instance, they use pathways like G protein coupled receptors pathways. So it's pretty cool, right? That's the main connection is like, do you need a separate method to tell the inside of the cell to do something? Like we think of steroid hormones, they're primarily nonpolar. They just diffuse right through the plasma membrane. They have a receptor that's in the nucleus or the cytoplasm. They just go straight to the source, right? They don't need a G protein coupled receptor. They don't need like a handoff, like a relay race, right? Like they just go and do it themselves, right? They still have a receptor, but where you'd find that and what it does is very different. So kind of cool little connection to a topic that we covered. Endocrine was a while ago, actually.
It was a while ago, yeah. Like when we did like like the whole cleavage idea, like when it came to embryogenesis, that was like maybe a few episodes ago, maybe like I don't know, maybe like five or ish episodes. Endocrine, endocrine was like a while ago. So go check it out. Just Google it wherever you are. Just like search endocrine JWMCAT podcast. You can find it. And this idea, hopefully, this idea, this connection between hormones and signaling cascades is a huge connection because this is one where a lot of times people memorize these facts like, oh, peptide hormone causes signal cascade, oh, steroid hormone does not cause, but they don't know the why behind it. But if you understand, oh, a steroid hormone doesn't need to, bind to something on the outside because it can just go right in, of course it's not going to use a GPCR. And this is what we kind of talk about all the time, the difference between memorizing and understanding. Because this is such a, I think, a common MCAT kind of question where it's like, oh, we've got a steroid hormone here. What kind of a receptor is it going to use? It's going to say something like, oh, a membrane-bound receptor or a GPCR. And there'll be some esoteric receptor you've never heard of as the only fourth option, but it's the only one that's not a membrane-bound receptor. And that's going to be your answer. I've seen those kinds of questions all the time where it's like the understanding these things are so important for scenarios like that. So I love this connection. It's such a huge one.
I love that you bring that up because 9 times out of 10, if that pops up, you know that most students are going to see that and be like, "Oh, I missed it because it's a content thing. I should have known what that random receptor was. Like I didn't have that memorized. I didn't do content enough. I need to stop doing practice and go back to my content phase." Solve it without the content first, or solve it with the content you have first. Okay. Only when there's literally nothing that you can do with your reasoning, with passage info, with the content that you have, should you assume that it's a content issue that created the problem. Okay. So just because you bring that up, if three are a cell membrane receptor and one is not, odd one out, right? Do your best. Um, Now, that being said, right, so we talked about the general context for G protein coupled receptors. We talked about the general flow, but we actually have different variants, and these are worth knowing for the MCAT. Okay, so there are a couple major types of G protein coupled receptors, but what's really cool is the whole cascade, the beginning part, the actual receptor itself, and for the most part, G protein layout, alpha, beta, gamma subunit, that's the same. What's different and what makes these pathways lead to completely different downstream responses inside of the cell is actually the G alpha subunit that you have. Okay, we have different types of G alpha, and keep in mind, G alpha is what's taking the action. So, what we see is you can have very different types of responses for a given receptor depending on what type of alpha you have, G alpha. Okay, so let's talk about the adenylyl cyclase pathway that, as you described, this is the main pathway that you're going to know. There are actually two different types of G alpha that you want to know. You're going to see GS and GI. Okay, this means G stimulatory, meaning it's going to stimulate adenylyl cyclase, and you have G inhibitory, which is going to inhibit adenylyl cyclase. Okay, so the pathway that goes with adenylyl cyclase and cyclic AMP, you have two alphas, one that activates, one that inhibits. Okay, GS and GI. Now, GS, okay, this is probably like the classical G protein coupled receptor pathway, right? Like most of the time, this is what we're talking about. The general flow, okay, you're going to have a ligand bind the receptor. Okay, that receptor is going to be associated with that G protein. G alpha is going to exchange GDP for GTP. It's going to become activated. It's going to dissociate. It is going to go off and activate adenylyl cyclase. Okay, adenylyl cyclase's actual job is to take ATP and make cyclic AMP. Okay, that's the key messenger created, so to speak, right? Cyclic AMP is then going to go on and activate what we call protein kinase A. Now, as it sounds, this is a kinase that targets a protein, okay? And it actually can have a variety of different proteins that it targets, okay? But when cyclic AMP is produced, it's going to activate this protein kinase and it is going to go off and phosphorylate a whole bunch of target proteins. Those proteins typically become activated through phosphorylation. Not always. They could be turned off. I don't know. Depends on the pathway. But that is what's going to create the internal or intracellular response. Okay, G inhibitory on the reverse end inhibits adenylyl cyclase, which means less cyclic AMP will be produced. Okay, that's the major difference. Again, depending on the type of G alpha you have, you will see differences in cyclic AMP production or adenylyl cyclase activity or protein kinase A activity. Again, you want to know the general flow for stimulatory, I would say, GS, and then just know inhibitory is the opposite.
Exact opposite, right? And I always like to remember, like, it's adenylyl cyclase, right?
Mhm.
Cyclasing, it's ATP turn, like taking that ATP and like cyclizing it to make cyclic AMP. Cyclic AMP, you'll a lot of times see it as C AMP, like a little lowercase c and an AMP. So if you ever see this lowercase c business going on, oh, that's cyclic AMP, there might be a hint that there's some G protein coupled receptor business going on. That's a really, really big hint there in the passage. And this is also where we see that amplification once again, where one cyclic AMP can activate multiple protein kinases A. And each one of those will activate phosphorylate multiple targets. Where again, one CMP, cyclic AMP, can do have a large impact. And we create multiple of those cyclic AMP with adenylyl cyclase. Really just trying to reiterate the fact of this amplification. This really, really large and very rapid response. Right? This is a very fast response, this thing. Whereas inhibitory, GI, just think the exact opposite. Yeah, it's inhibiting that adenylyl cyclase. I think this is where we can talk about some really good real-world applications of these G protein coupled receptors. I think the first one I think that's kind of an easy, quick one, which is epinephrine. Epinephrine, we know, has a very rapid response, right? Where, the adrenaline, if you ever had an EpiPen, this is a very quick response where it binds these beta adrenergic receptors. Now, again, the MCAT doesn't focus too much on, which kind of receptor it does it bind, but what we do need to know is that these receptors are GPCRs, right? And that means these receptors are associated with its own little G protein. And this is a case where we have a stimulatory G protein, where we have a stimulatory G protein targeting adenylyl cyclase, which ups cyclic AMP and causes all these effects. And the impact of many, right, with epinephrine is increased heart rate, increased blood pressure. These sorts of things are all that signal is caused by a G protein cascade. And again, big idea here, it's very fast. It's very fast and it's very rapid. That epinephrine, again, one of our amino acid hormones, right? This is one that is targeting in this way. Again, we want to reiterate, like, again, there's a really big connection with hormones when it comes to our signaling cascades. So a great reminder. And I know this next one, actually, I know you really want to talk about this next example, Molly.
It's just cool,
It's
okay?
very cool.
So there's another example here of cholera toxin. Now cholera, you may have heard about, right? It's a very, very serious disease, right? That causes severe diarrhea that could, prolonged diarrhea can lead to death, right? Like it's extreme and it tends to come on very quickly and create, again, very, very strong impacts on the individual. Now, what's interesting is cholera toxin actually acts through a G protein coupled receptor as well. Okay, so there is a G protein coupled receptor that is coordinated with actually a CFTR channel, which you may have heard as part of cystic fibrosis, which is actually how it gets its name. CFTR is all about chloride transfer, right? And we know in the lungs, which we won't talk about this too much, but in the lining of our, actually, I should just say our respiratory tract, not specifically our lungs, we have CFTR that helps loosen up mucus, right? We're basically allowing chloride to leave the cell because water will follow through osmosis and it helps us loosen up the mucus. And ultimately, with our mucociliary escalator, it helps get all the gunk out of our respiratory tract. CFTR is actually active in a lot of other places too, but namely, it's activated or it's active in our digestive tract. Now, what's really interesting about cholera toxin is it actually will bind to the G protein coupled receptor and create a very different impact on that G alpha. Okay, rather than having it exchange GDP for GTP. Now, keep in mind, we'll talk about this more in a moment, it's a GTPase, right? It's going to turn itself off because its job, the G alpha, is to cut that GTP into GDP. Once it's bound to GDP again, it's off. Okay. This actually it changes the functional group that's present on G alpha, making it constitutively active or constantly active. Now, this G alpha, after being stimulated by cholera toxin, is chronically on, right? This is a G stimulatory, right? So we have chronically elevated cyclic AMP because we have activated adenylyl cyclase again and again and again and again and again. This activates that CFTR membrane, which again allows chloride to leave. Water follows from osmosis. All of this water is being not pumped, I shouldn't use this word, but flowing freely into the digestive tract, which is creating, again, severe watery diarrhea, causing dehydration, which again can lead to death. It's potentially fatal disease, right? If not treated very quickly. So, very interesting, not cool because it's lethal, but it's interesting and it's really messing with this GPCR pathway, which I think is it's interesting to hear about, right? How high impact this really can be.
And it's really great that we bring this up, not because cholera toxin is MCAT content. For confidence, cholera toxin is not MCAT content, but this is how we'll see it in passages. They're gonna give us a pathway like this and say, oh hey, here's the pathway, right? This, oh, GTPase, oh, they switch, now it can't get rid of its GTP, now it's constitutively active, right? They can now ask us questions like, oh hey, if adenylyl cyclase is now always active. What do you think is going to happen to the amount of cyclic AMP? What do you think is going to happen to the amount of pKa and phosphorylation? Right? It's understanding, oh, that these are downstream impacts. When we change things, what's the impact, right? That's not something we can just memorize. We need to understand the impact of every change. Oh, maybe we switched up instead. Oh, what if we had, oh, gotten rid of the receptor altogether? Oh, would the presence of cholera toxin have an impact? Well, okay. Well, now maybe the cholera toxin itself wouldn't have an impact, but now why don't we have a GPCR? That probably has different impacts as well, right? These are the sorts of questions that now we want to be prepared for, but we won't be prepared for it by just memorizing GPCR, G protein, adenylyl cyclase, cyclic MP. Understanding the why and seeing it in these different places, that's how we can understand it. So it's a very, very, very cool, this pathway.
I agree. Yeah, so we talked about GS, G stimulatory, and GI, which is G inhibitory. That was all with adenylyl cyclase and cyclic AMP, which again is like the classical sort of pathway. It's the main one you want to know. There is another one, right? And I want to briefly mention it. It's something I want to be on your radar, but I don't think it's tested as frequently, and I don't think you need to know the same degree of depth, right? Because I would know those GS, GI very well, right? The other one is called GQ, right? And it's this pathway that basically is very related to phospholipids. Okay, so with GQ, which again is a variant of the G alpha subunit, rather than activating adenylyl cyclase when it subs out GDP for GTP, it's actually going to activate what we call phospholipase C, which you can imagine it sounds like phospholipid plus ACE as an enzyme, right? So its job is to cut a phospholipid. Now, these are not just any random phospholipids that you have in the membrane. It's not a random one. These are specifically phospholipids used for signaling. Okay, so the one that we look at, and again, individual names don't matter quite as much, but it's PIP2. Okay, so phospholipase C is going to cleave PIP2 into IP3 and DAG. Now, again, individual names, hopefully those don't show up. That would be pretty mean. It'd be a pretty narrow question if it did, but these are just modified phospholipids, very specific for cell signaling. And these downstream products are going to cause the release of calcium from the endoplasmic reticulum. Okay, calcium is therefore going to actually go on and carry out the downstream signal. And typically this is through PKC. So it's a similar origin in the G protein coupled receptor becoming activated by a ligand and then activating a G protein. That G alpha is going to dissociate, carry out some downstream function, but just a different flow, so to speak. And you'll see a different downstream effector or response.
Right. I think when it comes to me, like I like to kind of focus on the fact phospholipase C. Oh, therefore calcium. Right. That's a big
Oh
thing.
yeah,
Yeah, that's like a really big connection where it's like, oh, calcium is the effector, right? It's kind of like the cyclic AMP because it causes just a lot of calcium impact. Oh, and that calcium causes a lot of like impacts like cyclic AMP does.
and pKa is associated with adenylyl cyclase, even though that's before it's after adenylyl cyclase. But yeah, you're right. I like that.
Yeah, calcium. So I just
I
like
like
thinking
it.
of C's, calcium, different pathway than adenylyl cyclase, which is all about AMP and that sort
I
of
like
thing.
it.
That wasn't prepared before, by the way. That was just a great little mind connection there. So, but again, like I really want to reiterate, yeah, Molly, you're totally right. Like this is a pathway. Know that it exists, but adenylyl cyclase, that's the main one. GS, GI, cyclic AMP, PKA, that's the main one. Um, I think now would be a good time to make a little bit of a callback to last episode, because
I did. Mike loves this stuff, you guys.
I love vision.
I am
I love
sad.
eyes. I love eyes because it. I was so upset when I first learned about eyes. Like I remember when I studied about them, and I'm like, why would the eyes be like this? Because everything I would expect to be the case wasn't the case. And if you're like, oh my goodness, vision and eyes, what are they talking about? Go check out last episode if you want a little bit of context. But we like last episode we talked about, like the neural pathways, how we go from light to actual electrical signal and retina and all that thing to the occipital lobe. But one thing we talked about was that in the retina, we have these photoreceptors. And you would think, oh, light activates photoreceptor. Wrong. That's not what happens. You would think that, oh, light activates photoreceptors and that's what I see. No, no, no. Complete opposite, in fact. Photoreceptors are always on, but we didn't talk about last episode why they're always on. So, in these photoreceptors, we have our rods and our cones. And normally these are kind of constitutively active because they have a lot of cyclic GMP in them. And they're kind of similar to, again, it is a GPCR pathway, and they have, instead of cyclic AMP, it's cyclic GMP, right? And this cyclic GMP is acting on sodium channels and keeping them very open and very active. Very similar to our to action potentials, where these open sodium channels allow sodium in. When you're allowing sodium in, you're depolarized, you're active. The depolarization causes neurotransmitter to be released. So this is when rods and cones, there's no light at all. It's just active. Lots of cyclic GMP, lots of sodium coming in the cell, lots of neurotransmitter, specifically glutamate, being released. And that glutamate is inhibiting other things. So it's kind of keeping everything off, but by being on. Now, here's where it gets kind of weird. When light comes in, light, just a photon of light, hits a protein known as rhodopsin. Rhodopsin's the receptor. Because normally, remember when we talked about before, GPCR, there has to always start with a signal. But signal does not always mean molecule. Signal could be anything like light. Light is the signal that hits the rhodopsin. Rhodopsin's the receptor, the G protein coupled receptor, right? And by rhodopsin being hit, it changes its structure, just like we talked before. Changes the receptor and activates transducin. Transducin is the G protein. That's the G protein, and it's specifically an inhibitory G protein. And because now it's going to do the exact same things, it's got a GDP on it. It's become active. It's going to trade that GDP for a GTP. Alpha subunit, it's going to break off. Beta gamma, they're going to do their thing. Alpha subunit, though, is then going to activate a phosphodiesterase. So before we had adenylyl cyclase making more cyclic AMP. Now, in this case, we've got a phosphodiesterase. I told you before we had a lot of cyclic GMP. Phosphodiesterase, it's going to cut all of them. It's going to be cutting them, cleaving all of those cyclic GMP because they were cyclic. Now we're just cutting the ring. Now they're not going to be active. By light hitting that rhodopsin, that will decrease the amount of cyclic GMP in the cell. Again, because they're all getting cut. Less cyclic GMP means those sodium channels are not going to be on. They're going to close, and therefore sodium's not going to come into my cell anymore, and therefore it's not going to be depolarized. It's going to be hyperpolarized, and essentially turning off my photoreceptor. It was active, releasing a lot of glutamate, which is inhibiting downstream impact, but now we're turning it off. Now it's stopped, releasing neurotransmitter. But this is what actually causes you to see is the photoreceptors turning off that allows you to see, not them turning on, which I was like, what do you mean? But that's just how it is. Why is it? I'm sure there's an evolutionary biology reason for why light inactivates rods and cones, and that's why you see. But this was always something I was always very upset about, but why it's something that's very passionate, where I'm like, I don't know who made the rules, but maybe we should maybe we should change them. But yeah, that's and a good connection. If you're like, oh my goodness, Mike, you're talking about vision, and I'm like, what are you talking about? Go check out the last episode to get a little bit more context on vision. Then if you want, if you have already seen it, then now this all makes sense.
I love how passionate you get about these things, because it is so backwards. It's like, we think of G protein coupled receptors. It's backwards compared to what you'd normally expect, right? And the whole idea of a signaling cascade, even, it's backwards, right? Like if you're used to detect light, you'd want to be active when the light's there, right? No, you are less active when the light is there. You're more active when the light is not there. It's totally weird. It's totally backwards. And I'm sure there is an evolutionary biology reason. I don't know what it is. Shocker. I have no clue. I'm sure it
For anyone who knows Molly, that's a real shocker.
I'm sure it originated a very, very, very long time ago, probably in like the deep sea. Who knows? I couldn't tell you, but I'm sure it's old because it's bizarre, but it's cool, but it's bizarre. So I'm glad that we did that callback, especially because really we are going to move from vision into other senses, which, by the way, coming up, we will be covering additional senses that all act through primarily G protein coupled receptors and signaling cascades. So pretty fun. But again, just to take it back to where we started, right? Like we talked about the general flow, right? This idea that you have a signaling molecule on the outside of the cell and it needs to create a signaling response from the inside of the cell, right? And it's basically this elaborate game of like tag, like just activate this and then activate this and then activate this and then activate this and activate this. A lot of students are like, why the heck would you do that? What's the point? Why not just have molecule bind receptor, receptor activates everything else downstream? And you got to think, again, call back to what we were saying before about like cell division, right? That exponential growth. Each one molecule that's activated can typically activate a couple downstream targets. Okay, so one, let's say G alpha, might activate multiple adenylyl cyclases, or one adenylyl cyclase might activate or create multiple cyclic AMP, which then can activate multiple PKAs, etc. Right? Like every extra step in this pathway has the capacity to activate multiple downstream products, which means when you activate a GPCR coupled pathway, not only does this move fast, but it's very powerful, strong signal, right? Which is why, again, call back to endocrine system. We talk about how peptide hormones and, again, most amino acid derived hormones are short acting, but they are powerful compared to steroid hormones, which tend to take longer to kick in, but actually last longer as well. It has to do with this exponential amplification that we see with these. And with all this being said, especially if we talk about exponential amplification, it sounds like a positive feedback loop. We have to be able to turn it off, right? Turning the signal off is a very important portion. And yet we already really talked about it, right? G alpha is a GTPase. To become active, it has switched out GDP for GTP. I'm now holding on to this high energy GTP. But after a while, right, it's almost like it's got a timer, right? It's holding on to GTP and it's loving it, it's carrying its activity, but eventually it's going to inactivate itself by cutting that phosphate off. right? It's using the GTP. It's cleaving it, it's creating GDP, and now it's inactive again. Okay. That that prevents the continued activation of adenylyl cyclase, which then prevents the additional generation of cyclic AMP. But typically, we're going to go in and actually destroy cyclic AMP, right? Similar to what you were talking about with the vision pathway, Mike, right? We have a phosphodiesterase. So go in and cut up the cyclic AMP. It's not just enough to turn off G alpha. You got to get rid of some of that accumulated downstream product. And that's going to turn the signal off once it's been active. There's a lot that you could talk about with like sensitization to certain receptors or tolerance with continued activation, which is probably beyond the scope of what you'll be tested on, could be in a passage. There's interesting connections to even like drug tolerance and things like that, but we'll let that come up in a passage and you'll see it, right? They'll give you the logic that you need. Again, I really think that we've covered all the really core components of signaling cascades. Have we missed anything, Mike?
I honestly I don't think so. We've talked about the signal itself, how it can look very different. Could be a molecule, could be something like light, where it doesn't. We've talked about the receptors. Again, the receptors themselves are not the G protein. We've talked those are the kind of things that activates the G protein that to cleave and get active. We've talked about the downstream impacts of that G protein. It could be adenylyl cyclase. It could be something like a phosphodiesterase in light. It could be phospholipase C. We've talked about how those things create secondary messengers, or right, cause some other impact or some effect. And then we've talked about now we've wrapped up with how do we turn this off? And this again, this is a great connection back to cholera, where it's like, hey, cholera wasn't turning off because we changed this, right? GTP hydrolysis is what ends this. It's kind of like the thing that kicks it off. But if you stop the hydrolysis, you can't cut that GTP. You're just going to always be on. But in order for, to be in homeostatic balance, which our body loves to be in, we need to be able to turn off signals once the appropriate amount has been sent. And so now that's the whole signal response. And then maybe the body's going to wait for the next signal and for the next one. Like again, light hits our eyes all the time. We can't always have active rods and cones or deactive rods and cones. We have to sometimes have them on when you close your eyes. They're going to be on. When you open your eyes, they're going to be off. These kind of succession of these signals. This is how we detect different things. And this is actually going to be related very much to our next episode, where we're going to be kind of really diving because, like, I think if anyone's who's been listening for the last few episodes, we've been on this really like big sensation kind of topic where we hit some kind of topic, then we go back to sensation, then we hit another topic that's really important, then we go back to a sensation. Last episode we went on vision and optics. Now we're on more of a chemistry biology topic. We're going to go back to sensation and talk about smells and tastes next episode because they were like so related to this. Why are you thinking like what, Mike, Molly, why are we talking about signaling because cascades before that? It's because I think Molly mentioned this before. Smell and taste really rely on these pathways, on these receptors, and so that now anyone who's listening to this whole episode, you are now so prepared for a conversation in the next one.
I know. I cannot wait, especially to cover those together because a lot of people really they overestimate their taste buds, right? Like, oh, taste, it's all from the mouth and the tongue. Mm-mm. Most of your understanding and perception of flavor comes from your sense of smell, right? Your taste buds are remarkably simple. So I'm excited to talk about them in general, but also to talk about them together. Not only are they so related in how they individually function, but also they work together to dictate really your sense of flavor or your perception of flavor. So I'm very excited. This will be a fun one coming up. Thank you guys so much for listening. We hope you found this insightful and interesting. um, have a wonderful rest of your day or week or train ride or plane ride or wherever we are catching you. And we can't wait to see you in the next one.
Happy studying,
Happy
everyone.
studying. Oh, Jinx.